Welcome to our exploration of genome-wide association studies, or GWAS. These studies have revolutionized how scientists uncover the genetic factors behind complex traits and diseases. By scanning the entire genome, GWAS allows researchers to identify genetic variations associated with specific conditions. In this video, we'll journey through the basics of GWAS, its methodology, and its impact on modern genetics. Whether you're a student, a scientist, or just curious about how our genes shape us, you'll find the world of GWAS both fascinating and insightful. Let's begin by understanding what GWAS really means and why it matters. Genome-wide association studies are large-scale investigations that scan the genomes of many individuals to find genetic markers linked to specific traits or diseases. Unlike traditional genetic studies that focus on one or few genes, GWAS examines millions of genetic variants across the genome. This approach enables scientists to discover associations that were previously hidden, offering a broader and more comprehensive view of genetic influences. By comparing the DNA of people with and without a particular trait, researchers can pinpoint genetic differences that may contribute to that trait. GWAS relies heavily on big data. Researchers collect DNA samples from thousands, sometimes even millions of people. This massive scale is crucial because genetic differences that influence traits are often subtle and require large sample sizes to detect. Advanced computational tools and statistical methods are used to sift through the data, searching for patterns and associations. The more data available, the greater the power to uncover meaningful genetic links, making GWAS a cornerstone of modern genetic research. The process of a GWAS begins with collecting DNA samples from two groups, individuals with a particular trait or disease and those without. Researchers then use genotyping arrays to scan millions of single nucleotide polymorphisms, or SNPs, across the genome. Statistical analyses are performed to identify SNPs that occur more frequently in one group than the other. These SNPs are considered associated with the trait, providing clues about the underlying genetic architecture. Single nucleotide polymorphisms, or SNPs, are the most common type of genetic variation among people. Each SNP represents a difference in a single DNA building block, called a nucleotide. While most SNPs have no effect on health, some can influence how individuals develop diseases or respond to drugs. GWAS focuses on these tiny differences, searching for SNPs that are more common in people with a particular trait or condition, helping to unravel the genetic basis of complex traits. Common design in GWAS is the case control study. Here, researchers compare the genetic makeup of individuals with a disease cases to those without it controls. By analyzing differences in SNP frequencies between the two groups, scientists can identify genetic variants that may increase or decrease disease risk. This approach has been instrumental in uncovering genetic factors for conditions like diabetes, heart disease, and various cancers. Another important GWAS design is the cohort study. In this approach, a group of individuals is followed over time, and their genetic data is collected alongside information about their health and lifestyle. Researchers track who develops certain traits or diseases and look for genetic differences that might explain these outcomes. Cohort studies provide valuable insights into how genes and environment interact over a person's lifetime. GWAS, finding a true association between a genetic variant and a trait, requires rigorous statistical analysis. Because millions of SNPs are tested, the risk of false positives is high. Researchers use strict thresholds for statistical significance, often requiring a p-value of less than 5x10^8. This ensures that only the most robust associations are considered, reducing the chance of misleading results. One of the most recognizable visuals in GWAS is the Manhattan plot. This graph displays the results of millions of SNP tests across the genome. Each dot represents a SNP, and the height indicates the strength of its association with the trait. Peaks in the plot highlight regions of the genome with significant associations, guiding researchers to areas worth further investigation. Discovering an association in GWAS is just the first step. To ensure findings are genuine, researchers must replicate results in independent populations. Validation helps confirm that the association is not due to chance or specific to one group. Only associations that hold up across multiple studies are considered reliable, strengthening the evidence for a true genetic link. Linkage disequilibrium, or LD, refers to the non-random association of genetic variants in a population. In GWAS, LD can cause nearby SNPs to show association with a trait, even if only one is truly causal. Understanding LD patterns helps researchers fine-map genetic regions and identify the actual variants responsible for a trait. Once a region of the genome is associated with a trait, researchers use fine-mapping techniques to pinpoint the exact variant responsible. This involves analyzing additional genetic data and using statistical models to narrow down the list of candidate variants. Fine mapping is crucial for moving from association to understanding the biological mechanisms involved. After identifying associated variants, researchers perform functional annotation to determine their biological roles. This process involves integrating data from various sources, such as gene expression and protein function studies. Functional annotation helps link genetic variants to specific genes and pathways, providing insights into how they influence traits and diseases. Many traits and diseases are polygenic, meaning they are influenced by many genetic variants, each with a small effect. GWAS has been instrumental in revealing the polygenic nature of traits like height, intelligence, and risk for common diseases. By aggregating the effects of many variants, researchers can estimate an individual's genetic risk or predisposition for certain traits. Polygenic risk scores, or PRS, are calculated by summing the effects of many genetic variants associated with a trait. These scores can predict an individual's likelihood of developing certain diseases, such as heart disease or diabetes. While not perfect, PRS offers a glimpse into personalized medicine, where genetic information guides prevention and treatment strategies. GWAS has transformed our understanding of common diseases like diabetes, heart disease, and cancer. By identifying genetic variants that increase or decrease disease risk, researchers can uncover new biological pathways and potential drug targets. These discoveries pave the way for better prevention, diagnosis, and treatment options for millions of people worldwide. While GWAS is powerful for detecting common variants, it is less effective for rare variants due to limited statistical power. However, advances in sequencing technologies and larger sample sizes are making it possible to study rare variants. These rare genetic differences can have large effects on disease risk and are an important area of ongoing research. Most early GWAS focused on populations of European descent, limiting the generalizability of findings. Today, researchers are working to include more diverse populations in GWAS. Greater diversity improves the accuracy of genetic discoveries and ensures that all groups benefit from advances in genetic research. GWAS raises important ethical questions such as privacy, consent, and the potential misuse of genetic information. Researchers must ensure that participants' data is protected and that findings are used responsibly. Ongoing dialogue between scientists, ethicists, and the public is essential to address these challenges and build trust in genetic research. The rise of direct-to-consumer genetic testing has brought GWAS findings to the public. Companies now offer genetic risk reports based on GWAS data, allowing individuals to learn about their ancestry and health risks. While empowering, these services also raise questions about accuracy, interpretation, and the need for professional guidance. GWAS has contributed to the field of pharmacogenomics, which studies how genetic differences affect drug response. By identifying variants that influence how people metabolize medications, researchers can help doctors choose the right drug and dose for each patient. This personalized approach improves treatment effectiveness and reduces side effects. Traits and diseases are shaped by both genes and environment. GWAS can help identify gene-environment interactions, where the effect of a genetic variant depends on lifestyle or environmental factors. Understanding these interactions is key to developing effective prevention and intervention strategies tailored to individual needs. Epigenetics refers to changes in gene expression that do not involve alterations to the DNA sequence. GWAS findings are increasingly integrated with epigenetic data to better understand how genetic variants influence traits. This combined approach provides a more complete picture of the biological processes underlying health and disease. Discoveries from GWAS can inform gene editing efforts, such as those using CRISPR technology. By pinpointing genetic variants linked to disease, researchers can target specific genes for editing. While still in early stages, this approach holds promise for treating genetic disorders and advancing precision medicine. Despite its successes, GWAS has limitations. It often identifies associations rather than causation, and many discovered variants explain only a small fraction of trait heritability. Additionally, GWAS may miss rare or structural variants and can be confounded by population structure. Researchers continue to develop new methods to address these challenges and improve the power of genetic studies. One of the puzzles in genetics is the missing heritability problem. GWAS often finds that identified variants account for only a small portion of the genetic contribution to complex traits. This suggests that many genetic factors remain undiscovered, possibly due to rare variants, gene-gene interactions, or limitations in current methods. To address the complexity of genetic traits, researchers are integrating GWAS with other omics data, such as transcriptomics, proteomics, and metabolomics. These multiomics approaches provide a more comprehensive understanding of how genes, proteins, and metabolites interact to influence health and disease. GWAS findings are increasingly being translated into clinical practice. By identifying genetic risk factors, researchers can develop new diagnostic tools, therapies, and preventive measures. Translational research bridges the gap between genetic discoveries and real-world health benefits, bringing the promise of personalized medicine closer to reality. Success of GWAS depends on collaboration among scientists, institutions, and countries. Large consortia and data sharing initiatives have enabled researchers to pool resources and analyze data from diverse populations. These collaborative efforts accelerate discovery and ensure that genetic research benefits everyone. Open science and data sharing are essential for maximizing the impact of GWAS. By making data and results publicly available, researchers enable others to validate findings, conduct new analyses, and build on previous work. This culture of openness fosters innovation and speeds up the pace of genetic discovery. The future of GWAS is bright, with advances in technology, data analysis, and international collaboration driving new discoveries. As sample sizes grow and methods improve, researchers will uncover more genetic factors and gain deeper insights into the biology of health and disease. The integration of GWAS with other scientific fields promises even greater breakthroughs ahead. Precision medicine aims to tailor healthcare to individual genetic profiles. GWAS plays a key role by identifying genetic variants that influence disease risk and treatment response. As our understanding grows, doctors will be able to offer more personalized care, improving outcomes and reducing unnecessary treatments. Educating the public about GWAS and genetics is vital for informed decision-making. Outreach programs, accessible resources, and transparent communication help demystify genetic research and encourage participation. Engaged communities are more likely to support and benefit from advances in genetic science. GWAS not only informs medicine but also sheds light on human evolution. By studying genetic variants across populations, researchers can trace the history of traits and adaptations. This evolutionary perspective deepens our understanding of how humans have adapted to diverse environments and challenges. GWAS is not limited to humans. Researchers use similar approaches to study genetic traits in animals and plants, improving agriculture, breeding, and conservation. These studies help identify genes responsible for important traits such as disease resistance or yield, benefiting food security and biodiversity. Technological advances such as next-generation sequencing and high-throughput genotyping have fueled the growth of GWAS. These innovations make it possible to analyze more genetic data faster and at lower cost. As technology continues to evolve, the potential for new discoveries in genetics expands. Machine learning and artificial intelligence are increasingly used in GWAS to analyze complex data and identify patterns. These tools can uncover subtle genetic associations and predict disease risk with greater accuracy. The integration of AI promises to accelerate genetic discoveries and enhance our understanding of health and disease. Large biobanks, such as the UK Biobank, store genetic and health data from hundreds of thousands of participants. These resources are invaluable for GWAS, providing the scale and diversity needed for robust discoveries. International collaboration among biobanks enhances the power and reach of genetic research. GWAS findings are paving the way for personalized prevention strategies. By understanding an individual's genetic risk, doctors can recommend lifestyle changes or screenings tailored to their needs. This proactive approach helps prevent disease before it starts, improving health outcomes for everyone. Interpreting GWAS results can be challenging. Many associated variants are located in non-coding regions of the genome, making it difficult to determine their function. Researchers use a variety of tools and databases to predict the impact of these variants, but much work remains to fully understand their roles. GWAS has the potential to transform public health by identifying genetic risk factors for common diseases. These insights can inform screening programs, guide resource allocation, and shape health policies. As genetic research advances, its impact on population health will continue to grow. Bioinformatics is essential for managing and analyzing the vast amounts of data generated by GWAS. Specialized software and algorithms help researchers process genetic information, identify associations, and visualize results. The field of bioinformatics continues to evolve, supporting the growing demands of genetic research. Community involvement is crucial for the success of GWAS. Engaging participants, respecting cultural values, and ensuring transparent communication build trust and encourage participation. Inclusive research practices help ensure that genetic discoveries benefit all communities. As we look to the future, GWAS will continue to play a central role in unraveling the genetic mysteries of health and disease. With ongoing advances in technology, data analysis, and collaboration, the possibilities are endless. By working together, scientists and communities can unlock the full potential of genetic research for generations to come. Genome-wide association studies have transformed our understanding of genetics and disease. From uncovering risk variants to guiding personalized medicine, GWAS continues to open new doors in science and healthcare. As research progresses, the insights gained from GWAS will help shape a healthier, more informed future for all. Thank you for joining us on this journey through the fascinating world of genetic discovery.